Dear students, this is 12th zoology video class, chapter 3, reproductive health, higher secondary, second year zoology, part 8. Part 8. Last class we have seen what is assisted reproductive technology, what are the technology I use in ART. So these are the topics we have seen. Next topic is the one surrogacy. Next topic is the one surrogacy is a methods of assisted reproductions. Surrogacy is a method of assisted reproductions or agreement whereby a woman agrees to carry a pregnancy for another person who will become the newborn child's parent after birth. Though in vitro fertilization, that is IVF, embryos are created in a lab and are transferred into surrogate mother's uterus. I'll show you one picture. Here, this picture, you can see couples are there, one male and female are there. So these couple do not have chance of carrying, the woman do not have chance to carry young ones because of her health issue. So for that, they are just selecting other women to carry their embryo. So for this woman name only we call as a surrogacy. That is what we are saying it. Surrogacy is the methods of assisted reproductions, or where a woman agrees to carry a pregnancy for another person. That is we call as a surrogacy. Here, who will become a newborn child parents after birth. Though through in vitro fertilizations (IVF), the embryos are created in a lab and transferred to the surrogate mother's uterus. This is what we call as a surrogacy. The next topic is the one male infertility. Azoospermia is defined as the absence of spermatozoa. Azoospermia is defined as absence of spermatozoa, which means spermatozoa means the formations of sperm we call as spermatozoa. Zoospermia means the absence of sperms we call as spermatozoa. In ejaculate semen on at least 2 occasions and is observed approximately 1% of populations. Microtesticular sperm extractions. This is a procedure or methods where the sperms can extract. That is we call as a microtesticular sperm extraction. TESE. They'll ask you expand, underline one word. Microsurgical sperm retrieval from the testicle involves a small midline incisions in a scrotum. With the help of this microtesticular sperm extractions, just they'll insert one needle in the scrotum. Scrotum means what? That's a testis, where testes are covered by a sac-like structure, that is because of scrotum, through which one or both testicles can be seen. Under the microscope, the seminiferous tubules are dilated, which means expand. Dilator means expand, and small amount of testicular tissue in an area active sperm productions are removed and improved for sperm yield compared to traditional biopsy techniques. So they'll take out they'll insert one needle in the scrotum and just they extract the testes, the sperms from the testes. They extract the sperm with the help of that syringe and instruments. That is we call as a TESE. Then here, next topic is the one detection of fetal disorder during early pregnancy. If any child undergoing for disorders, any disease, that can be detected by ultrasound scanning. What is ultrasound scanning? Ultrasound has no known risk other than mild discomfort due to pressure from the transducer on the abdomen or vagina. Even if you just take ultrasound scan means no known risk. Okay, unknown risk can be identified in this one. No radiations used during this procedure. Ultrasonography is usually performed in the first trimesters for dating. When the first 3 months of child develop, during that time only this ultrasonography are used to perform. Determinations of the number of fetuses and how many child are formed for that one also determinations can be found with help of ultrasound scanning. And for assessment of early pregnancy complications, if the child, whether they have any complications, any disease in the body or not, we can identify with help of this ultrasound scanning one. The next topic is the one amniocentesis. Amniocentesis involves taking a small sample of amniotic fluid. Amniocentesis involves taking a small sample of amniotic fluid that surrounds the fetus, surrounds the child, to diagnose for chromosomal abnormalities. Here you can find. Here this is the child. The child just developed one fluid. That fluid name called as a amniotic fluid. Can you find? They injecting the needle in the amniotic fluid. They extract that fluid. In that fluid, where we can identify the abnormalities of chromosomes. The chromosomes, therefore, there are 46 chromosomes will be there in a human being. Whether that 46 chromosomes are normal or abnormal, where we can identify by extracting this amniocentesis. Here, these are the parts you can look at. Placenta, fetus means the child, developing child in the uterus, and this is the structure of uterus. So that is because of amniocentesis. Okay, then here you can look at the picture. The next one here, continuations of this amniocentesis is generally performed in a pregnant woman between 15th and 20th week of pregnancy by inserting a long thin needle through abdomen into the amniotic sac to withdraw a small sample of amniotic fluid. That's what just we are seeing. One needle just injecting in the abdomen where they can extract the amniotic fluid. The amniotic fluid contains cell shed from the fetus. Okay, what are the cell sheds that can be identified with the help of this amniotic fluid? The chorionic villus sampling. Chorionic villus sampling, CVS, is a parental test that involves taking a sample of placental tissues to test for chromosomal abnormalities. So chorionic villus sampling in order to identify chromosomal abnormalities, whether the chromosomes of 46 chromosomes are growing properly, do they have any abnormalities, any disorders. With help of this CVS, where we can identify the chromosomal abnormalities. The next one is the one fetoscope. This fetoscope is used to monitor the child, is to monitor the fetal heart rate. Fetoscope is the one to check the child heart rate and other functions during late pregnancy and labor. Okay, late pregnancy and labor, after pregnancy, where they can identify the fetal heart rate. The average fetal heart rate is between 120 and 160 beats per minute. An abnormal fetal heart rate pattern may mean that the fetus is not getting enough oxygen, and it indicates other problems. If the child does not get a proper heartbeat, about 122 and 160 beats per minute, which indicates the child does not get enough oxygen and they have some problems. So that is the fetoscope. One way they'll ask you what the use of this fetoscope is in order to identify fetal heart rate. What is the average fetal heart rate? Means 120 or 160 beats per minute. So one way they'll ask you. And with this, just we have completed this chapter 3. And fetoscope, continuations of fetoscope is here. Yeah, handheld Doppler device is often used during prenatal visit to count the fetal heart rate. Here, the fetoscopy to identify the fetal heartbeat. There is a prenatal before the child gets detached from the mother's uterus. There is an instrument handheld Doppler device. It's a device where they can use very often during prenatal before the child birth, before the child born, the visits to count the fetal heart rate with help of this Doppler device, handheld Doppler device. During labor and continuous electronic fetal monitoring is often used. So here there is a small information which are there. Vitamin E is known as an anti-sterility. One was the last key important one, which helps in normal functioning of reproductive structure. Vitamin E, it is anti-sterility, helps in the normal functions of reproductive structure. Then sex hormones were discovered by Adolf Butenandt. One word, important one word. 11th July is observed World Population Day. 11th July is observed World Population Day. 1st December is observed World AIDS Day. One word, all these things they'll ask you. What is NACO? NACO, National AIDS Control Organization, was established in 1992. Syphilis and gonorrhea are commonly called as international diseases. International diseases. And there is a small information bits here. Breast self-examinations and the early diagnosis of cancer. You can go through this one and you can understand. There is a mammogram are done for women above the age of 40 years. For young girls and women below the 40 years. Ultrasound of the breast aids in early diagnosis. The breast cancer can be identified. That's a small information which are given. You can go through. And today we have completed chapter 3 fully. If you have any doubt, you can contact me. Today we have completed what is surrogacy, what is male infertility prevention, what are the detections of fetal disorder during early pregnancy we have seen. So these are the topics we have. Detections of fetal disorder during early pregnancy we have seen. Then what is ultrasound scanning we have seen. And what is the functions of amniocentesis we have seen. Then also we have seen what is CVS. Also we have seen what is fetoscopy. And also we have seen here the confirmations of fetoscopy. You can go through small informations. With this, just we'll wind up our class. If you have any doubt, you can contact me through WhatsApp. Okay, bye. Take care.